Imagine a world where the waiting list for organ transplants is a thing of the past. Welcome to a new dawn in biotechnology. Every day we inch closer to this reality, with researchers at Harvard University leading the charge. They've developed a new hydrogel ink infused with gelatin fibers, a breakthrough that's paving the way for the 3D printing of functional heart muscle. Picture this, a ventricular heart muscle, crafted from this specialized ink that beats just like a human heart. This isn't just a static model, but a dynamic, living tissue that maintains its 3D shape post-printing, enabling the alignment and synchronized beating of cardiomyocytes. These are the cells that are crucial for heart muscle contraction. The key to this innovation? A spinning jet technique that creates microfiber materials, eliminating the need for electric fields which may degrade certain proteins. This technique allows researchers to create a 3D printed ventricle that, while not as strong as real heart tissue, can pump significantly more fluid than previous 3D printed heart chambers. The implications of this breakthrough are wide-reaching. We're in an era where over 100,000 people are waiting for an organ transplant in the United States alone, and every day, 17 people die waiting. But what if we could print organs on demand? What if the waiting list for organ transplants became obsolete? This technology has the potential to revolutionize organ transplantation. By creating personalized living structures, we could see improvements in treatments ranging from skin grafts to cancer research and beyond. This is not just science fiction. It's a reality being forged in the laboratories of Harvard University. But how does one print a heart muscle? Let's dive into the heart of the matter. At the core of this groundbreaking technology is a newly developed hydrogel ink. This isn't your typical pen ink. It's a unique substance infused with tiny gelatin fibers. These fibers are the secret ingredient that provides the gel with its special properties. After the printing process, the gel maintains its three-dimensional shape. This is crucial, as it allows the printed structure to resemble the actual organ as closely as possible. But it doesn't just stop at looking like a heart muscle. The gelatin fibers also play a vital role in enabling the alignment and synchronized beating of cardiomyocytes. Now, if you're wondering what cardiomyocytes are, they're essentially the cells that make up your heart muscle. They're the tiny powerhouses that contract rhythmically, creating that familiar heartbeat we all know and love. The gelatin fibers in the hydrogel ink align these cardiomyocytes and enable their synchronized beating. This is a huge leap forward in bioprinting, as it's the first time we've been able to recreate this essential function of the heart muscle. However, developing this hydrogel ink was no small feat. It required an intricate understanding of both biology and materials science. The researchers had to ensure that the gelatin fibers were the right size and concentration to support the cardiomyocytes and mimic the extracellular matrix of the heart. This is the network of proteins and other substances that surround cells and provide them with essential support and signals. This process of developing the hydrogel ink and creating a 3D printed heart muscle is a delicate dance of science and engineering. It requires precision, patience, and a deep understanding of the human body. But the result is a functional ventricular heart muscle that beats just like a human heart. This process is intricate and delicate, much like the heart itself. A spinning jet technique, you say? Now, that's interesting. Indeed, it is. The spinning jet technique is an innovation in biotechnology that's taking the world by storm, and it's a crucial component in the development of the 3D printed heart muscle. But what exactly is it? Well, imagine being able to create microfiber materials without the need for electric fields. That's precisely what the spinning jet technique does. It's a method that eliminates the need for electric fields, which can degrade certain proteins, thereby ensuring that the structural integrity of the biomaterials is maintained. This is an essential step in the process because it allows the 3D printed ventricle to maintain its shape post-printing. This is where the gel with fiber infusion, or GIF, comes into play. The GIF is a type of hydrogel ink infused with gelatin fibers. When combined with the spinning jet technique, it enables the alignment and synchronized beating of cardiomyocytes, the cells that are critical for heart muscle contraction. But how does it work? The spinning jet technique spins the hydrogel ink at high speed, creating a stream of microfibers. These fibers are then collected and used as the building blocks for the 3D printed heart muscle. This method allows for the precise control of fiber diameter and alignment, which is critical for mimicking the natural structure of heart tissue. In essence, the spinning jet technique is like a high-tech loom, weaving together the fibers of life. It's a fascinating process, with the potential to revolutionize not just heart transplants, but the entire field of organ transplantation. The spinning jet technique is a game changer, paving the way for more breakthroughs in biotechnology. It's a testament to the incredible strides we're making in science, and a glimpse into a future where organ transplants are more efficient, more reliable, and more accessible to those in need. We're living in an exciting time, with innovations like the spinning jet technique taking us closer to a future where no one has to wait for a life-saving organ transplant. It's not just about creating a beating heart, it's about giving hope and life to countless individuals around the world. And that, my friends, is truly heartwarming. The heart is a pump, and the 3D printed ventricle is no different. Every beat of our heart pumps blood throughout our body, supplying the oxygen and nutrients that our cells need to function. It's a complex process that involves the contraction of cardiomyocytes, the cells that make up our heart muscle. In the world of biotechnology, researchers at Harvard University have printed a 3D ventricle that can mimic this process. This isn't just any 3D printed ventricle, but one that can pump significantly more fluid than its predecessors. This feature is crucial in making the 3D printed heart muscle more functional. Picture, if you will, earlier 3D printed heart chambers. While they were impressive feats of biotechnology, their function was limited. They could hold fluid, sure, but could they pump it? Not quite. Their capacity to mimic the natural pumping action of a heart was limited, to say the least. Enter the new 3D printed ventricle. Infused with gelatin fibers, this ventricle isn't just a static structure. It's a dynamic organ capable of pumping a significant amount of fluid. This isn't just a step forward in 3D printing technology. It's a leap towards creating functional artificial organs. The 3D printed ventricle still has a way to go. It's not as strong as real heart tissue. It can't pump as much fluid, but it's getting there. With each beat, it's getting closer to mimicking the real thing. With each pump, it's proving that the dream of creating artificial organs isn't just a dream. It's a reality that's within our grasp. Though not as strong as a real heart tissue, the 3D printed ventricle is a significant leap forward towards creating artificial organs. From the realm of science fiction to the brink of reality, the 3D printed heart muscle is transforming the future. The pioneering work of the researchers at Harvard University has opened a new chapter in the annals of biotechnology, bringing us one step closer to the day when organ transplants may not be limited by donor availability. This breakthrough in 3D printing technology has the potential to revolutionize healthcare. Imagine a world where the wait for a heart transplant could be significantly reduced. A world where a patient's own cells could be used to create a perfectly matched organ, eliminating the risk of rejection that haunts current transplantation procedures. These advancements could also redefine the landscape of medical research. Scientists could create and study human tissues in the lab, accelerating our understanding of disease and speeding up the development of new treatments. Consider the potential impact on drug testing. Instead of relying on animal models, researchers could use bioprinted tissues to test new medications, leading to safer and more effective therapies. Furthermore, the development of a functional 3D printed heart muscle could pave the way for similar advancements in the creation of other organs and tissues. This could usher in a new era of personalized medicine, where treatments are tailored to the individual patient's unique biology. And let's not forget the profound societal implications. This technology could alleviate the suffering of thousands of patients waiting for organ transplants, transforming lives and families. While we are still in the early stages of this journey, the progress made so far is promising. The challenges are significant, but so are the potential rewards. The 3D printed heart muscle serves as a potent symbol of what human ingenuity can achieve when fueled by the desire to improve and save lives. With each beat of the 3D printed heart muscle, we step closer to a future where organ shortages could be a thing of the past. The future is here, and it's beating with life.